Mitosis and meiosis are two very important processes of cell division that occur in our bodies every day. Before either can occur, interphase must first take place. Interphase, which accounts for 70% of the cell cycle, is divided into G1 and G2 for cell growth and S for chromosome replication. Extended fibers of DNA form a tangled mass of chromatin. RNA synthesis takes place in the nucleoli, and the centrioles will have also replicated. To better explain this process from a more interesting perspective, I will compare the centrioles to two pairs of people. They will be on two boats, the centrosomes, fishing on a lake, the cell itself, and surrounding a school of fish, the duplicated chromosomes. Fishing line can represent the mitotic spindle, while floating logs around the fish can represent a nuclear envelope. Mitosis can now proceed. In mitosis, a cell separates the replicated chromosomes in its nucleus to produce two identical daughter cells. The first phase is prophase, where the chromatin condenses into chromosomes and the nuclear membrane breaks down. Each duplicated chromosome appears as two sister chromatids joined together at their centromeres. A mitotic spindle attached to the centrosomes begin to form composed of microtubules and asters, and the centrosomes move away from each other. On a lake, the two fishing boats have moved apart and cast lines into the water to begin snatching fish, which have moved closer together. The second and longest phase of mitosis is metaphase. The centrosomes are now at opposite poles of the cell and the mitotic spindle forms. Chromosomes line up along the metaphase plate with each double-stranded chromosome attached to a spindle microtubule at its centromere. In other words, the two fishing boats have moved to opposite ends of the lake and have each caught a fish from every pair of fish on two separate lines. The fish have lined up along the lake center to be equidistant from both boats. The third and shortest phase of mitosis is anaphase. Anaphase begins when the centromeres of each chromosome separate, creating two identical sister chromatins per pair. Each chromatid has a centromere and a sessile chromosome, creating twice as many chromosomes as there were in metaphase. Spindle microtubules pull the chromosomes by their centromeres toward opposite poles while elongating the cell. On the lake, the fishermen have pulled a fish from each pair towards their corresponding boat by shortening the fishing line just like a mitotic spindle. By the end, each pair of fishermen will have an equal number of fish. The final phase of mitosis involves telophase and cytokinesis. The poles now each have the same number of chromosomes as the parent cell. The nuclear membrane reforms, nucleoli reappear, and the chromosomes become less condensed. Next, cytokinesis divides the cytoplasm and its contents. In animal cells, the cell is pinched in two by a cleavage furrow. In plant cells, the cell plate is formed by vesicles. For the fishing analogy, the fishermen have changed their minds and released the fish from their lines, allowing them to swim around like uncoiled chromatin. It has also been very rainy outside, causing the lake to grow in size, overflow, and split across its center to create two identical lakes, like the two-end diploid daughter cells that result from mitosis. Mitosis is a very important process for growth, and any error can cause cancer or other cellular mutations. In meiosis, the number of chromosomes per daughter cell is cut in half from the parent cell due to two cell divisions. This process, divided into meiosis 1 and meiosis 2, is very important for sexual reproduction as it creates genetic variation among the four daughter cells. Like mitosis, it is preceded by the replication of chromosomes. The meiotic phases of prophase, metaphase, anaphase, telophase, and cytokinesis are similar to those in mitosis with slight differences. In prophase 1, chromosomes condense. Unlike in mitosis, replicated homologs pair up along synapses and crossing over DNA segments occurs at chiasmata. The replicated homologous chromosomes will be represented by different colored fish. The homologous pairs are arranged as tetrads on the metaphase plate in metaphase 1, and not as individual chromosomes as in mitosis. Also unique to meiosis 1 is how the sister chromatids remain attached while the homologous chromosomes separate in anaphase 1. The sister chromatids in mitosis had separated. At the end of telophase 1 in cytokinesis, each cell has a 1N haploid set of replicated chromosomes. Similarly, the fishermen have an equal number of fish but an unequal number of type. In meiosis 2, the two cells produced in meiosis 1 undergo another division to separate the sister chromatids. First, a spindle reforms in prophase 2 so that by metaphase 2 the chromosomes are positioned on the metaphase plate. The two sister chromatids on each chromosome are not genetically identical, so that when they separate in anaphase 2, 4 haploid daughter cells are produced at the end of telophase 2 in cytokinesis. The chromosome number has now been doubled from meiosis 1. Each of the 4 daughter cells is genetically distinct from the other daughter cells and from the parent cell. Meiosis 2 can be compared to 2 more fishing boats joining the first 2 on the lake, and then further dividing the amount of fish so that each of the 4 boats now has a different combination of fish. Hopefully this presentation of mitosis and meiosis as a comparison to fishing was entertaining and helped to clarify any confusion between the two. Thanks for watching.